Welcome to Praadis Institute of Education.
Biotechnology, Principles and Processes. Hello children, today we are going to learn about the principles and processes of biotechnology. Topic 1. Introduction and Principles of Biotechnology. Introduction. The technique of using live organisms or enzymes from organisms to produce products and processes useful to humans is called biotechnology. Many processes like in vitro fertilization leading to test tube baby, synthesizing gene and using it, developing a DNA vaccine or correcting a defective gene are also parts of biotechnology. The European Federation of Biotechnology (EFB) has given a definition of biotechnology that comprises both traditional and modern molecular biotechnology. The definition is as follows: the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. Principles of biotechnology. Among many, the two core techniques that enabled the birth of modern technology are genetic engineering, techniques to alter the chemistry of genetic material, DNA and RNA, to introduce these into host organisms and thus change the phenotype of the host organism. Bioprocess engineering, maintenance of sterile, microbial contamination-free ambience in chemical engineering processes to enable the growth of only the desired microbe or eukaryotic cell in large quantities for the manufacture of biotechnological products like antibiotics, vaccines, enzymes, etc. Traditional hybridization leads to inclusion and multiplication of undesirable genes along with the desired traits. This problem can be overcome by using the technique of genetic engineering. The techniques of genetic engineering allow us to isolate and introduce only one or a set of desirable genes without introducing undesirable genes into the target organism. The piece of DNA, when integrated into the genome of the recipient, may multiply and get inherited along with the host DNA. An alien DNA is linked with the origin of replication of host DNA so that this alien piece of DNA can replicate and multiply itself in the host organism. This can also be called as cloning or making multiple identical copies of any template DNA. Stanley Cohen and Herbert Boyer in 1972 isolated the antibiotic resistance gene by cutting out a piece of DNA from a plasmid autonomously replicating circular extrachromosomal DNA of Salmonella typhimurium. The cutting of DNA at specific locations became possible with the discovery of the so-called molecular scissors, restriction enzymes. The cut piece of DNA was then linked with the plasmid DNA. These plasmid DNA act as vectors to transfer the piece of DNA attached to it. The enzyme ligase acts on cut DNA molecules and joins their ends. This makes a new combination of autonomously replicating DNA created in vitro and known as recombinant DNA. When this DNA is transferred into E. coli, it could replicate using the new host DNA polymerase enzyme and make multiple copies. The ability to produce multiple copies of antibiotic resistance gene in E. coli was called cloning of antibiotic resistance gene in E. coli. Recombinant DNA technology, or also called genetic engineering, deals about the production of new combinations of genetic material artificially in the laboratory. These recombinant DNA, rDNA molecules are then introduced into host cells where they can be propagated and multiplied. Steps of recombinant DNA technology: identification of DNA with desirable genes, introduction of the identified DNA into the host, maintenance of introduced DNA in the host and transfer of the DNA to its progeny. Tools of recombinant DNA technology. Following are the key tools of recombinant DNA technology: restriction enzymes, polymerase enzymes, ligases, vectors, and the host organism. Restriction enzymes. In the year 1963, the two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated. One of these added methyl groups to DNA while the other cut DNA. The latter was called restriction endonuclease. The first restriction endonuclease, HIN2, always cut DNA molecules at a particular point by recognizing a specific sequence of 6 base pairs. This specific base sequence is known as the recognition sequence for HIN2. Restriction enzymes belong to a larger class of enzymes called nucleases. These are of two kinds, exonucleases and endonucleases. Exonucleases remove nucleotides from the ends of the DNA, whereas endonucleases make cuts at specific positions within the DNA. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. Palindromes are groups of letters that form the same word when read both forward and backward. For example, Malayalam. As against a word palindrome, where the same word is read in both directions, the palindrome in DNA is a sequence of base pairs that reads same on the two strands when the orientation of reading is kept the same. For example, the following sequences read the same on the two strands 5' to 3' direction. This is also true if read in the 3' to 5' direction. 5' 𝐭𝐴𝐴TTC 3', 5' 𝐭𝐴ATTC 3'. Restriction enzymes cut the strand of DNA a little away from the center of the palindrome sites, but between the same two bases on the opposite strands. This leaves single-stranded portions at the ends. There are overhanging stretches called sticky ends on each strand. These are named so because they form hydrogen bonds with their complementary cut counterparts. This stickiness of the ends facilitates the action of the enzyme DNA ligase. When cut by the same restriction enzyme, the resultant DNA fragments have the same kind of sticky ends, and these can be joined together end-to-end using DNA ligases. Separation and isolation of DNA fragments. The cutting of DNA by restriction endonucleases results in the fragments of DNA. These fragments can be separated by a technique known as gel electrophoresis. Since DNA fragments are negatively charged molecules, they can be separated by forcing them to move towards the anode under an electric field through a medium or matrix such as agarose, which is a natural polymer extracted from seaweeds. The DNA fragments separate, resolve according to their size through sieving effect provided by the agarose gel. Hence, the smaller the fragment size, the farther it moves. The separated DNA fragments can be visualized only after staining the DNA with a compound known as ethidium bromide, followed by exposure to UV radiation. The separated bands of DNA are cut out from the agarose gel and extracted from the gel piece. This step is known as elution. The DNA fragments purified in this way are used in constructing recombinant DNA by joining them with cloning vectors. Cloning vectors. Plasmids and bacteriophages are commonly used vectors for cloning. They have the ability to replicate within bacterial cells independent of the control of chromosomal DNA. Following features are required to facilitate cloning into a vector. A. Origin of replication, ORI. It is the sequence from where replication starts, and any piece of DNA, when linked to this sequence, can be made to replicate within the host cells. This sequence is responsible for controlling the copy number of the linked DNA. B. Selectable marker. Help in identifying and eliminating non-transformants and selectively permitting the growth of the transformants. Transformation is a procedure through which a piece of DNA is introduced in a host bacterium. Generally, the genes encoding resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline, or kanamycin, etc. are considered useful, selectable Markers for E. coli. C. Cloning sites. In order to link the alien DNA, the vector needs to have single recognition site for the commonly used restriction enzymes. Presence of more than one recognition sites within the vector will generate several fragments which will complicate the gene cloning. The ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistance genes. For example, you can ligate a foreign DNA at the BAMH1 site of the tetracycline resistance gene in the vector PBR322. The recombinant plasmids will lose tetracycline resistance due to insertion of foreign DNA but can still be selected out from non-recombinant ones by plating the transformants on ampicillin-containing medium. The transformants growing on ampicillin-containing medium are then transferred on a medium containing tetracycline. The recombinants will grow in ampicillin-containing medium but not on that containing tetracycline. But non-recombinants will grow on the medium containing both the antibiotics. In this case, one antibiotic resistance gene helps in selecting the transformants, whereas the other antibiotic resistance gene gets inactivated due to insertion of alien DNA and helps in the selection of recombinants. Alternate selectable markers have been developed which can differentiate recombinants from non-recombinants on the basis of their ability to produce color in the presence of chromogenic substrate. In such a case, a recombinant DNA is inserted within the coding sequence of an enzyme alpha-galactosidase. This results in inactivation of the enzyme. It is called insertional inactivation. The presence of chromogenic substrate gives blue-colored colonies if plasmid in bacteria does not have an insert. Presence of insert results into insertional inactivation of alpha-galactosidase and no color is produced by the colonies. Such colonies are identified as recombinant colonies. D. Vectors for cloning genes in plants and animals. Bacteria and viruses have been transferring their genes to transform eukaryotic cells in order to force them to do what the bacteria or viruses want. Now, these tools of pathogens can be transformed into useful vectors for delivering beneficial genes to humans. For example, the tumor-inducing TI plasmid of Agrobacterium tumefaciens has now been modified into a cloning vector which is no more pathogenic to the plants. This vector can now be utilized to transfer beneficial genes into many plants. Competent host for transformation with recombinant DNA. DNA is a hydrophilic molecule and cannot pass through cell membranes. In order to force bacteria to take up the plasmid, the bacterial cell needs to be competent to take up DNA. This is done by treating the bacterial cells with a specific concentration of a divalent cation, for example, calcium. It increases the efficiency with which DNA enters the bacterium through pores in its cell wall. Recombinant DNA can then be forced into such cells in the following steps. Cells along with recombinant DNA are incubated on ice. They are then briefly placed at 42 degrees Celsius heat shock. Then they are put back on ice. This is not the only way to introduce alien DNA into host cells. In a method known as microinjection, recombinant DNA is directly injected into the nucleus of an animal cell. In another method, suitable for plants, cells are bombarded with high-velocity microparticles of gold or tungsten coated with DNA in a method known as biolistics or gene gun. And the last method uses disarmed pathogen vectors, which when allowed to infect the cell, transfer the recombinant DNA into the host. Thank you.